So the problem that we're trying to solve is identifying unknown molecules. And this is just given a sample. We want to see what molecules are in there. This is not an easy task, but it is a very important task in many different areas for biological processes and many other areas like drug discovery, personalized medicine, environmental toxicology, and many more. So the tool that we use is tandem mass spectrometry. It is fast, it's sensitive, and it's highly reproducible. So the first and main challenge that we're trying to solve is that predicting the whole molecule from the spectrum is not easy. The spectrum is lossy. It doesn't always gather all the information that we need to go back from the spectrum to the molecule. Two relatively different molecules could potentially provide the same spectrum. So it is a bit lossy. And also, we don't have enough pairs of spectrum and molecule to train a machine learning model that helps us do this. Um, and One other thing that we can notice is that the domain for the potential molecule is too big. So it's not just a simple mapping that we need to train because the total number of molecules is too big. And unfortunately, we don't have enough spectrums and their pairs of molecules to train the model. So what we try to do is to learn the modifications from the analog. So instead of training this whole very hard problem, we want to do something that is simpler. And that is given a base molecule, given an analog to our unknown molecule, can we now predict what is the actual molecule? So instead of trying to solve this very hard problem, we're trying to solve a much simpler problem that we're trying to figure out the molecule structure given a base. So for example, if I give you this painting and ask you what it is, you might have some guesses, but you cannot be sure. But if I provide you with this base as well, and I tell you like, this is a painting, but it also like this part we know for sure exists in the final result. And then you put this together, now it is a much easier problem to solve, right? It is now much more clear that this is a photo of the Eiffel Tower. Um, so knowing that base helped us to figure out the final like painting a lot easier. And this is the case for machine learning as well. Trying to train a model that just trains the differences is much easier because it's a smaller space. So to this purpose, we use diffusion models. Now, just an overview for diffusion models. Assuming that these diffusion models are the exact same things you see in different applications such as ChatGPT, it's the baseline for many of the generative AI models that make pictures, and I'm sure you've seen examples, if not used them yourselves. So in those models, you just pass a prompt. For example, give me a picture of a dog, and then uh, you need this image. But what is a good image? Like, what happens if the model gives you this image? Is that an image of a dog? Well, of course, it's not. It's a useless image. It's just pure noise. What if the model gives you this image? It's an object. It's not just what you ask. And then what if you get this? It's close. It's not a dog, but it's very close, right? And then this is great. This is a dog. But what about this? Like two dogs. Is that what you asked for? And so quantifying if the model is working good or not, it's not a trivial task. Figuring out if the output is a good image or not to train your model, this is not easy, right? So for the diffusion models, instead of just trying to annotate if the image is good or not, we want to learn the probability distribution of the data set. So assume that we have this whole, like this is a representation of the whole possible images. Assume that this is like all the pixels in an image. Um, we know that some parts of these whole probability distribution area is where the images of the dogs exist, right? So for example, imagine that like this is just an illustration, right? But imagine that golden retrievers are coming from these areas, and then German shepherds are coming from these areas, right? So we want to learn which part of our probability distribution is the parts that like correspond to the images of the dog. Later on, we can use this to generate new images of the dog. So the way diffusion models work is that given the sum of the samples, so here you can see some of the samples of the data, for example, images of different types of dogs. We gradually add noise to these data until we get to something that is pure noise or very close to pure noise. Now, this is called the forward process. Now, in a reverse or backward process, we try to learn how to denoise each of these steps. So we want to go back from a more noisier version to a bit denoised version. Now, if we can train a model to do this denoising for us, then for sampling or for generating images of whatever we're looking for example, the dog, we can just start from pure noise and gradually denoise it until we end up to a point within our distribution that corresponds to the prompt that we were asked. So if you look at this image, for example, we started from the training set, we gradually noised it to get to this like random distribution, right? And then we trained the model in the backward process. And now if you choose one sample like the red dot and you just gradually denoise it, because the model is trained to move those random noises into somewhere in the data that we're interested in, then we end up with an image in the part that we're interested in. Now, you can add more condition to this backward process. For example, you can add a text, for example, a dog driving a car, and then just condition your model based on that as well. So for Modistruct, the core idea is if we have the spectrum of an unknown molecule, so we have the spectrum, the mass spectrum, the tandem mass spectrum of that unknown molecule, and we want to see what it is. But we also have two more additional information. One is the spectrum of a known substructure. So a molecule that is substructure of the unknown molecule. Here you can see an example of a substructure molecule and the unknown molecule. And also, so the spectrum and the structure of these like substructure, we want to use this substructure molecule and then predict the unknown molecule from that. So we know the base and we just want to inpaint around it, just like the Eiffel Tower image that we saw earlier. So let's first look at an actual run on the Modistruct, where it just does this gradually denoising steps until it reaches the final version, which is the complete denoised version. And you can see as the time progresses, it is matching more to the substructure and the actual final result. And once it is done with denoising, we can see the actual final result for the unknown molecule. Now, how does it work? So we're specifically for this task, we're using a discrete version of diffusion models. And the first step is to create an adjacency matrix. Now, I'm not sure if you're all familiar with adjacency matrix, but here's a quick overview. So for this graph, which is a representation, a 2D representation of a molecule, we first create the matrix. So if node 1 is connected to node 2 with, for example, a double bond, then we add this value to between node 1 and node 2 in the matrix. And then if this is a single bond between node 1 and node 7, we add that to the matrix. And then we continue doing this until we have this adjacency matrix. Now, we use this in our forward and backward process. So the noising happens on this adjacency matrix. So at each step, we add noise to this matrix, meaning that we might add some edge between two nodes or two atoms, or remove a bond between two atoms, or maybe change the type from single bond to double bond and things like that. So that's the noising process. And then during the denoising process, the model trains to just denoise from a step to a previous step. In this specific case, we want to add some conditions to it as well. So what are those conditions? We want to pass, so these are the conditions. We want to pass the substructure, right? We want to pass what is the base that the model has to inpaint around. Additionally, we want to pass the spectrum or the information we can gather from the spectrum. And that's like the text that guiding the diffusion models when generating the photos. Here, we want to guide our model with the spectrum. So we want to say, give me a model given the spectrum. Give me a model that matches this spectrum. So here are the information that we pass to the model. We pass the graph nodes, which we gather from running the series. I think we use the latest version of it too. And then y hat here is the fingerprint of the molecule. I'm going to talk about how we get the fingerprint of the molecule, but for now, assume that we already have the fingerprint of the molecule. And then we have the adjacency matrix, which we're trying to denoise one step. And then lastly is the mask for the substructure, just letting the model know where the substructure exists. And finally, where the modification sites are. This one, again, I'm going to talk about it in a bit. So the conditions that we're giving the model is the fingerprint, the modification sites, what the substructure is, and then the model is trying to denoise the edges based on that. And if you're not familiar with what the fingerprint of a molecule is, it's essentially a vector that just says whether some different structures exist in the molecule or not. Okay. So now the model has two parts. One is the encoder, which is going to give us this modification sites and the fingerprint, and then the decoder, which you're seeing here right now. So for the encoder part, we want to generate those two different things, right? The fingerprint and the modification site. And to do that, first, we want to encode the differences between two spectrums. We want to see if there is any meaningful information in the spectrum of this unknown molecule with regard to the spectrum of the known molecule. And to do this, we borrow the idea used in the TransXRM paper. So given the two spectrums, we're going to subtract the different peaks in the two spectrums and populate this matrix with the values. So we're checking, we're populating this matrix, and then we pass this matrix to a transformer encoder to encode each of the different peaks in the spectrum. And because we're looking at the differences, these encoded vectors for each of these peaks for the unknown molecule contains information on how much it changed regarding to the analog molecule. In another step, we also encode the spectrum of the unknown molecule. We want to just have some representation of the spectrum of the molecule as well. And to do that, we use the same formula transformer used in the MIST paper. So that was the first part that was encoding the spectrum. The second part is to encode the structure of the base graph. And for that, we're using a graph attentional network. And this is the final structure of our encoder. So the two spectrums, we encode them. the graph for the substructure, we encode that as well. And using these encoded vectors, we try to predict the fingerprint of the final molecule and also where the modifications happen. So given these informations, the model can predict the fingerprint and the modification sites. And then we use this information to pass to the decoder to figure out the structure of the final molecule. So one thing we saw was that even though we trained this Modistruct to provide a final molecule, we see that it is even better at predicting the fingerprint than MIST, which was one of the one of the models that we use as part of this module. So we can see that, excuse me, we can see that the idea of just predicting the changes instead of predicting the final result is something that is applicable in this area. So here we can see that Modistruct full is outperforming MIST. Another observation that we have is that training the model with the two different tasks in mind. So the two different tasks are modification sites and predicting the fingerprint. Training the model with the two tasks is improving the performance of the model compared to training the model without the modification site task. So even though we don't necessarily need the modification sites for the final result, we can just pass the fingerprint. That's an information we can gather from the spectrum. We can predict the fingerprint and then use that to predict the final molecule. We see that if we also try to predict the modification sites, then we get a better performance even in predicting the fingerprints. Another observation is that our model outperforms Modifinder in finding the modification site. So Modifinder is a paper specifically dedicated to finding modification sites between a molecule and its analog. And we see that Modistruct, even though it was not primarily trained on this task, it can outperform Modifinder by 4 times. And then finally, we can see that Modistruct paper is outperforming other models at predicting the final molecule. So we have 2 different criteria here. The first one is accuracy, and it says whether we were able to predict the final molecule correctly or not. And we can see that in 5% of the scenarios, for the mass spectrum dataset, which is specifically a very hard and curated dataset for this task, we're able to predict the correct structure. And then the second property that we're analyzing is the average tiny model. So we're checking if the tiny model of our predicted model versus the true model is better than the other models or not. And we can see that Modistruct is outperforming other models here as well. Yeah, that's it. Thank you.
Great. Any questions, people?
Yeah, thanks for the invite. And yeah, as you introduced, my name is Abzer. I'm a final year PhD student at Functional Metabolomics Lab in Tübingen University in Germany. Today, I would like to talk about the FBM and Stats app and the protocol, which mainly works for the statistical analysis of feature-based molecular networking results. And yeah, I guess most of the people here are already familiar with the feature-based molecular networking workflow. If not, I just wanted to give a quick introduction, where feature-based molecular network practically takes all these features or the metabolites, practically the MS-MS spectra, and cluster them based on their MS-MS spectral similarity. So this is a way with which we can make sense of the large number of metabolites, especially in terms of untargeted metabolomics. A huge issue is with annotation, and many of these metabolites are not annotated. In that cases, molecular networking is a great way to propagate through, to improve this annotation by propagating through these network. Thereby, we can putatively also annotate some of these metabolites or features. And for our work, we mainly use untargeted metabolomics, and molecular networking is particularly feature-based molecular networking is something that we very commonly do, and that is also one of the most popular networking workflow in GNPS. So how we do that is we get our data from mass spectroscopy, and we particularly use LC, MSMS, tandem mass spec. Reza already talked about it. And after we get our raw data, we use a feature detection tool such as MZMine, with which we get our MS1 level information, such as the peak area abundances, and also we kind of cluster the MS2 level information, the fragment spectra. So with both of this MS1 and MS2 level information, we could put it in the GNPS platform, particularly feature-based molecular networking workflow, which helps us build this molecular network and also helps us with annotation. We could also use annotation tools, in silico tools like Sirius, which also helps with chemical formula prediction, can also help a little bit with the annotation. So after we do this feature-based molecular networking is where the FBM and stats guide comes into play. So once you do the job, you can use the FBM and stats app or follow the protocol to perform some stats and data visualization. MZMine is something you locally install in your computer. GNPS is available online, and Sirius is kind of semi, where you install it locally, but also needs guest internet access to get the information from the server. And FBM and stats guide is available online, and it can also be downloaded for Windows OS. However, to also simplify this whole pipeline, especially for a beginner to untargeted metabolomics, one could simply use GNPS dashboard as well, which takes the raw data, also perform feature detection, from which one can run the molecular network job, and also use FBM and stats guide, the web app. And I've also added the web app links for this. And what we need to run this analytical pipeline is a feature quantification table, a metadata, and an annotation table. So feature quantification table is the table that we get from our feature detection tool, such as MZMine, that we use. So here we have the list of features, the features in rows, and and the samples in columns. And the numbers that you see are the peak area abundances of these features. And a metadata table is something that we can easily create in an Excel sheet. And this is practically describing all the samples in the feature table. So we give additional information, such as what are the different types of samples, groups, replicates, whatever the conditions that is useful. And as an output of GNPS, we also get annotation table that helps explain some of these compounds, what these compounds are. And we can also use additional information. We can also use such information from tools such as Sirius. So in the protocol, first we talk about some of the good practices and also some steps of how we can get from the raw data to the feature table. So we have the LCMSMS raw data, then we convert it from raw to MZML, a centroided format. And some of the steps we've described how one can, like the steps that involves in the MZMine feature detection tool, then how we go from there to obtaining, running the GNPS job. The actual steps of the web app or involves from here, the data pretreatment. So what we do essentially is that we pull the feature-based molecular networking results, and from there, we first perform some data cleaning, and then we do some stats. So for this data pretreatment or the data cleaning, we focus on 3 steps, blank removal, imputation, and some data transformation steps. For the first step, blank removal, we try to remove these features that could be potentially from blanks, solvent blanks, extraction blanks, etc. So ideally, we are trying to remove some of these background features. It already reduces the number of features. And after that, we try to impute. Here we try to replace the zeros with a minimal value and try not to introduce additional variation as well. So here we assume that these zeros are not true zeros, but maybe because the values were not detected because of the instrument's limit of detection. So yeah, zeros could also be problematic sometimes in some statistical tests such as PCA. So we also try to replace these zeros in imputation. And for the data transformation techniques, we can either go for normalization. The most commonly used is the TIC, total ion count normalization, or sample-centric normalization, where we try to normalize these features across samples. Or you can also do scaling, where we scale them or normalize them within each sample. Yeah. So after you do some of these data cleanup pre-treatment steps, then the user can go on to perform some multivariate or univariate stats. So for the multivariate stats, we provide some unsupervised and supervised techniques. For example, with the unsupervised, they can visualize their samples in using a PCA or PCOA plot. Most of you will be familiar with that. So here, what we are trying to do is that instead of like showcasing all the, hundreds of features, we are trying to reduce their dimensions into a 2D plot so that we can have a kind of a bird's eye view, trying to understand the underlying pattern in the sample. For example, here we see these 3 groups nicely in a PCOA plot. And yeah, we also provide clustering, heat map, etc. And for the supervised, we use a random forest technique where the data trains on the train set based on the metadata category that you use, try to understand the underlying pattern, and then we predict it on a test set. As a result of this, we get a list of important features. And for the univariate stats, this is, for example, like ANOVA or a t-test, where what we are essentially doing is trying to see what features are different among the different groups. Like if a particular feature is significantly higher or lower among these different groups, and you get the list of features that are either significant, non-significant, and also their p-values, so and so. So this is the type of test and analysis that we can do in the pipeline. And with this workflow, we published this last year as a Nature Protocols paper called Statistical Analysis of FBMN Results from Untargeted Metabolomics Data. And the FBMN Stats Guide is also an output of this protocols work. And yeah, the web app is a simple Streamlit web app. Started initially with Axel and me, and this web app can be easily accessed from the FBMN task page from GNPS, as you can see, downstream analysis, hitchhiker stats guide, or it is also hosted in the GNPS to a server that you can also access it through this URL online. In addition, this can also be downloaded to run on Windows OS. We don't have anything for Mac OS, but people can easily clone it from the GitHub and run it locally as well. So to download it for the Windows apps, for the Windows OS, you can download it from our functional metabolomics webpage under resources. And yeah, also want to mention that right now we are revamping the website, and Rithi, bachelor student in UCR, she's helping with that. So yeah. In addition, or to the web app, the protocol is mainly based on scripting languages, R, Python, Kind2, which is also popular in the microbiome community. And all of this is available in our GitHub functional metabolomics lab page. And these scripts, we provide them in Jupyter Notebook format, can also be accessed through Google Colab. Just wanted to show screenshot of the Jupyter Notebook. The cool thing about these scripting languages is most of the people would be also comfortable with scripting languages, or if they're willing to learn, want to learn, then they could use this. But then all these steps between these different notebooks are Python or Kind2 notebooks. It's all comparable, and that's how we've written in the protocols paper. And as I mentioned, just a quick decision tree to show to choose between notebooks and app. So if the user want to have a quick view of their data, of course they can use the FBM and Stats Guide web app because it's streamlined. You take your task ID of the web app, put it in the from the GNPS page, put it in the web app. We can quickly go through our results. But if you want more independence, or if you're already familiar with the scripting languages, then also if it's a larger data set, then of course the user can use either R or Python. Yeah. And not just the FBM and Stats app, I also wanted to talk a little bit about the other modular web apps which are available in the GNPS2 ecosystem. Right now, there's a preprint available by Helena, and where they talk about some of the cool apps, other apps that are also available in this ecosystem, collectively called as Metabo apps, as they all help to further explore from the molecular networking results. For example, like MassQL, where you want to look at a particular pattern. Let's say I want to go from my MS2 information of 130 MZ, so and so, then I can look for that particular pattern either within my own data set or across public data sets, or there are tools like MAST, which helps to look for repository scale investigations, where we query the MSMS spectra again within a set of data sets or like across public repositories, and try to contextualize the unannotated feature if it is from a tissue sample, biofluid sample, etc. And similarly, I also particularly wanted to talk a little bit about some of the apps that we do in our functional metabolomics lab. And also, these are my kind of PhD projects. So with FBM and Stats, this is particularly tailored for FBM users, but can also be used in any context, in other contexts as well. And the important thing about the tool is that it can get the data directly from the FBM job ID, this streamlining the process. And we do the stats, and we can also download all of these statistical analysis and can further, we can then add this information back to the molecular network, such as here. This is just like a hypothesis. The hypothetical example that I'm showing here is that molecular networking already gives us these nice clusters when we have not great annotation. Then we could already contextualize with the chemical classes or molecular family, saying that there's a carbohydrates group, alkaloids group, so and so. But with the stat information added to the molecular network, for example, here we could ask further questions like, why is this particular feature significant when all the other things are not significant? And what is the MC difference? And it just helps us to understand the data better. And other tools that are available are ChemProp2, which is a correlation-based scoring of these individual node pairs in the molecular network, adding directionality to these edges, particularly in the case of incubation experiments, when you have multiple time points and are looking for biotransformations, then ChemProp2 helps to prioritize some putative biochemical transformations. And moving beyond metabolomics, we also have choromics, which helps with multi-omics integration. For example, if you want to correlate your microbial features with the metabolite features, looking for co-occurrence pattern, or like anti-correlation or a negative correlation pattern. Yeah, with that, I just wanted to show a little bit of the Stats Guide app. Okay. Yeah, now I have opened it through the Stats Guide. We have our homepage where we already explain all these information, and with the data prep, we have multiple file origin. The user can manually input the data, quant table and the metadata table, or they can add the GNPS task ID. It could be from GNPS 1 or GNPS 2. This is from feature-based molecular networking. And we also show a couple other example data sets, and you could also use classical molecular networking bucket table just to understand the data. So now I will use an example task ID just to show probably PCA and a t-test. And here I'm just going to my GNPS2 ID, and I just, this is my task page. And here I'm just choosing a random job ID. So these are unique task IDs for each of these jobs. And going back again to my web page. I hope this works. Okay, so I'm just added the task ID, and the user can already visualize their quantification table, metadata, and the ID, M over Z, retention time information. They're all added together to provide a unique ID for each of these metabolites. And you can also go through the different metadata categories. Basically, we have two sample types here, skin sample and plasma sample. So each of these pages and all of these modules, they have these information section as well, either with visual illustration or with some textual information. So I just do blank removal. And here I'm just going to choose, and here you can also see your metadata table converted to a summary table. So here I can also nicely see there's my column, MZ-Mind sample type, which says that I have two sets of samples, blank and sample. And yeah, I have 84, this is not arranged properly, but I have 84 samples and 4 blanks. So I could just go here and choose my sample first, and can also have a quick preview. And it says that I have 84 samples. And again, I have to choose my blanks, which I would just go back choosing the blank 4 samples. And this is the cutoff threshold that I was talking earlier. So what we do is ratio-based cutoff. So we look at the mean value of each of these features for samples versus blank. And everything that is above this cutoff, we flag them as a noisy, like a background feature. So the higher the cutoff, then I will be retaining more features. Therefore, there could possibly be more noisy features as well. So if you reduce the cutoff, let's say I just want 0.1, that means I just want 10% like noisy features in my data, then that will also increase the number of noise features. So for example, if I reduce it, you could see that I also increase the noise features. And after that, I end up with a feature count of 3,200. So with that, I can also go to the imputation step. I'm going to just simply impute the missing values. And here you could see that these values will be filled with random number between 1 and 6. So 6 is the limit of detection for this data set. So I will choose just random values and fill it with this. And I'm just going to simply perform normalization, TIC normalization, and submit data for statistics. And all of these tables, of course, they can also be downloaded. And if the user want to analyze with their own script or anything, they could do that as well. But if you want to also try it out here, we also have many tests here. So if I go to the attribute sampling location, I guess, okay, not applicable, and upper back. Okay, so here we can already see that we see 88% of separation of this data was based on the attribute sample type, or like, I mean, is based on the x-axis, first axis, and we could see a nice separation between skin and the plasma samples. So we could also do OA, principal coordinate analysis, which is similar to PCA, but uses different distance matrices to calculate this distance between these samples. But I just want to show one univariate test as well. And with that, I'd be pretty much done. And here, if I choose attribute sample type, like a simple t-test, which looks at their mean and see if different among these two different groups, then it would call that to be significant. And if there is no difference among the mean of these two groups, then it would call it as non-significant. And so we have a feature significance plot, and then single metabolite plot, and finally the data. And what we can see here is that all these features in red, they're all significantly different. And some of these ones are different, I mean, higher in skin samples, and these one are higher in plasma samples, and the ones in gray in the bottom, they're non-significant, meaning that there was no big difference between the mean of these features. And user could also explore a single metabolite plot where they can see the box plot format. And we also indicate if it is a significant, if it is a significant feature or so. So they could also either type it in or go check a particular interesting feature. And finally, they could also get these information in tabular form so that they could again add this information, feed this information into the molecular network. For example, here you can see that all of these features, the first row is a combination or a concatenation of their unique ID, M over Z, and then retention time. So they could split it and use this unique ID information, combine it back to the molecular network node table, and they can visualize which features are significant or not significant within the molecular network as well. So yeah, this is pretty much what I wanted to show, but I also want to quickly, thank the people, both in Tübingen University, because the functional metabolomics lab is now in UCR, and Daniel is also here. So I want to thank both the members of the lab in Tübingen and in UCR, and also thanking the virtual multiomics community. Many of them helped with the protocols, with testing and providing useful suggestions, and also Axel and Riti, particularly for helping with the web app. And of course, my supervisor, Dr. Daniel Petras, and also Dr. Ming. And yeah, thank you all for listening.